My name is Jason Weckstein. I'm the curator of ornithology here at the Academy of Natural Sciences, and I want to welcome you to the ornithology collection here at the Academy. So what we want to do today is take you on a tour to show you where our specimens are housed, and then we want to tell you a few example stories that illustrate how we use specimens for our research. Our collection is among the largest collections of bird specimens in the world. We are the second largest university-based museum collection in the world, second to Harvard. And our collection includes approximately 220,000 specimens. This includes everything from some skeletons to bird skins like these. And I'm going to tell you a little bit about what we use these kinds of specimens for. And we even have specimens in alcohol that can be used to look at anatomy, internal anatomy. And then we have tissue collections here at the Academy. One of the beauties of natural history collections is that specimens like these that are housed in our collections can tell us all kinds of different stories. So you probably think, this is just a collection of dead things. Well, as a biologist, we actually see this as a living collection that tells us about life on Earth. This is the documentation of life all over our planet. And this is true of all of our collections here at the Academy. And each of these specimens is a lot like a book in a library. And just like in a library, every book has different words in it. Well, every specimen has different stories to tell us. And all of these specimens have specimen labels. This is an aracari, which is a small-bodied toucan. This is one of the groups of birds that my lab studies here at the Academy. And this happens to be from Ecuador, from the province of Esmeraldas. And there's all kinds of detailed locality information on this tag that tells us where this bird came from. It tells us it was a male. It tells us the latitude and longitude where it was collected, the date that it was collected, who prepared it, and then all kinds of information on the back that tells us about the internal structures on this animal that can tell us about whether it has bred before, what its age is, what its condition is. And then on something like a toucan where we have colorful bills, usually the preparator draws a picture of the specimen because that is information that's lost. The bill fades, unlike the plumage, which does not fade over time. Now, what do we use these specimens for? Well, what's exciting is that we use these specimens for an incredible array of projects. What that means is the person that was out in the field studying these things may have used these specimens for one project, but because they're housed here in the collection, they can be used for hundreds and hundreds of years. And because we have specimens that date back to even as far back as the late 1700s, our collection is incredibly valuable for looking at change over time. This might be change over time in the distributions of birds. It might be change over time in terms of how climate affects the distributions of birds, change over time in terms of disease that birds carry, all kinds of different questions that we can ask. What's exciting is that we are developing new tools every day, and these new tools allow us to unlock new information about these specimens. So one of the tools that we often use, and we're going to take you on a tour of our lab later, is DNA. We study DNA of these specimens, and DNA can tell us a huge, important story about all kinds of different pieces of the biology of these birds. It can help us understand the evolution of coloration, the evolution of migration. It can help us to understand whether or not different populations are different than each other. It can help us reconstruct the tree of life, basically a genealogy like your family tree. We do that with these bird specimens. And in modern days, we collect tissue samples, but in the old days, of course, we didn't know about DNA and tissue samples were not collected. But what we can do is we can cut a little piece of a toe pad off of the bottom of one of these old specimens. So we can go to specimens from the 1800s, for example, and we can go into a special lab that we have downstairs and we can extract DNA from that little piece of toe pad, and we can use that to tell stories about these old specimens that in the past we would never have been able to unlock that information. So what I want to do now is tell you a little bit about one of the many projects that we're doing in the ornithology department. So our group studies everything from the genealogies of birds to the diversity of parasites that live on and in birds. And if you've spent any time here in the mid-Atlantic, or really any time across North America, out bird watching in particular, you've probably seen this species. This is a sharp-shinned hawk. This flies over my neighborhood during migration, and they breed here in the state of Pennsylvania. These are small bird-eating hawks. And what's interesting about these is that our North American population of sharp-shinned hawks is a migrant population. They migrate south during the winter. However, there are populations in the tropics that don't migrate at all. And those include populations of birds in South America like these, which are sedentary, That means they don't migrate. They stay in the places where they breed all year long. And then there are populations on the Caribbean islands in Cuba, Hispaniola, and Puerto Rico. And what's interesting is that if you look at the scientific literature, these are all considered one species, and they're considered one management unit from a conservation perspective. But the populations in some parts of the tropics are not the same in terms of their size as the populations in North America. So the populations in North America are very healthy, and there are large numbers of these sharp-shinned hawks. But if you go to Puerto Rico, the numbers are extremely low on the island. And that's in part due to human habitat alteration and also hurricanes, and a combination of those two. And my group here at the Academy of Natural Sciences, including research associate Therese Katnack and PhD student Matt Halley, we are working with the Peregrine Fund to study the population genetics or the conservation genetics of this group. And what's exciting is that we found that these different populations of sharp-shinned hawks are genetically different than one another. And they're so genetically different that we now know that this North American sharp-shinned hawk is extremely different than the Caribbean forms, and that the different Caribbean forms on the 3 Caribbean islands, on Cuba, Hispaniola, and Puerto Rico, those are also genetically different enough that they should all be considered different species. Next, we're going to go to Camila Kuwabara, who's one of our PhD students from the Drexel Bees department, and she works here in the ornithology department. She's a member of my lab, and she's going to talk about some of the research that she's doing on parasites of birds.
My name is Camila Kuwabara, and I'm a first-year PhD student here at the Bees department. My interest is in chewing lice and their evolution with birds. You know that you might have lice, and also other organisms have lice, including birds. So I'm interested in studying them. So right now, what I'm doing here is to separate the organisms that were collected from the birds. So for example, here I can see 2 different types of parasites of birds. One is chewing lice and the other ones are feather mites. Over here, these are the adults of chewing lice. These are immatures. And over here, we can see a feather mite, very tiny.
Next, we're going to go to Nate Rice, who's our collections manager in the ornithology department.
My name is Nate Rice, and I'm the ornithology collection manager here at the Academy of Natural Sciences. I take care of our bird specimen collection, and I just want to give you some information about it as I walk through here. We have over 200,000 bird specimens in our collection, and that represents almost every species of bird in the world. The cabinets that you see that we're walking through here house one of the world's most important bird collections. Researchers from around the world request loans from our collection, and we send out about 70 loans every year to museums around the world. That represents thousands, sometimes tens of thousands of specimens that we loan to institutions for all sorts of interesting research, to answer all sorts of fascinating research questions. The collection is housed in over 170 of these steel white cabinets that you see here. And we have everything in size from the smallest hummingbirds to the largest vultures and everything in between. Our collections come from virtually every corner of the planet, and the collections predate the founding of the Academy. We have dated specimens to the 1790s, and we add several thousand new bird specimens every year from a combination of salvage work, of window-killed birds, and birds that hit cars or hit power lines, and then direct collecting from international collecting expeditions. All right, I'd like to show you a really interesting family of birds. These are the hummingbirds, family Trochilidae. And we have 2 cabinets, the one open behind me and then one to my left, my right, your left here. So these are the smallest birds in the world, and in fact the smallest bird in the world is in this family. Let's have a look at some of these here. So this is the biggest of the hummingbirds. This is giant hummingbird, found in the Andes of South America. and all hummingbirds are found in the New World. Here's the sword-billed hummingbird. This particular one was collected in Ecuador in 1992. One of the real charismatic hummingbirds, not that any of them are uncharismatic. This is the spatule-tailed hummingbird. This is an adult male. Lodogysia mirabilis is the Latin name. And this is the female here. These are found in Peru, small range size in Peru. And like I mentioned, the smallest bird in the world happens to be a hummingbird, and it's called the Cuban hummingbird or bee hummingbird. So this is the tray that it's on. These are Costa's hummingbirds and calliope hummingbirds. These are birds found in the United States, found in the southwestern United States. And then if we sneak back here, this is an adult male Cuban hummingbird or bee hummingbird. And you can see that it's much smaller than my fingers. In fact, it's only about a third the size of the label that's on it. This is an adult male, and this was collected on the Isle of Pines, January 29th, 1931, collected by a gentleman named James Bond. James Bond, most of you know him as a superhero international spy, but we know him as a world-famous, internationally renowned ornithologist who was a world's expert on Caribbean birds. And this is one of the many hundreds of birds that he collected in the course of his research here at the Academy. These are the fairy wrens, and this is a group of birds found in Australia and New Guinea. And we'll zoom in on some here. These are some birds that I collected in Australia in the early 2000s. And the really cool thing about our collection and the history of our collection is that laying right next to these birds collected in the 2000s are birds that were collected by John Gould on the first collecting expeditions to Australia in the 1830s. So that represents a huge temporal spectrum of the species on the continent of Australia. You'll notice that there's labels on each specimen, and each specimen has its own unique identifier number, and then there's massive amounts of data on these labels. All that label information and the catalog number are entered into a database, and you can find that information online through the Academy's website or on some data servers on the web. Okay, we had a look at some small birds. Now let's have a look at some of the huge birds in our collection. This is a cabinet that contains vultures, the New World vultures. So black vulture, turkey vulture, and then these are the condors. So these trays here are condors. This is Andean condor. This is a bird that was collected in 1936 in Ecuador. I'll put my hand in here for size so you can see that this is a monstrous bird here. Wingspan probably close to 10 feet, maybe 12 feet. But just like the fairy wrens that we looked at, the prep process is exactly the same. All of the insides are removed and just the skin and feathers are left, and then that's stuffed with cotton, and you can even see cotton sticking out where the eyeball used to be here. This is a really interesting object. It is called the hyoid or hyoid apparatus, and this is basically the tongue bone of this bird. You can see it has sort of a toothy feel to it here, and that's to help tear flesh that the bird would eat.
Next, we're going to go to Janice Despato, who's our lab manager. Janice is going to take us down into the lab to show you how we use DNA to study the parasites that live inside of birds.
Hi, my name is Janice Despoto. I'm the lab manager for ornithology. Here is where the magic begins. We study birds and their parasites. Luckily, bird blood is nucleated, which means that it has a lot of genetic material in it. So it starts with either a bird blood spot that's deposited on a filter paper called an FTA card, or we can also extract DNA from a tissue sample that was taken from a bird during an expedition. From the blood spot, we use molecular tools that will extract the DNA from the blood and lyse the cells to release the DNA in a process that takes about 18 hours of incubation in an incubator with shaking. So now that the cells are totally lysed, the DNA then is deposited on these little filters. Now each one of these circles represents one bird. So we can do, in essence, we can do 96 bird samples at a time here. Once the DNA has been washed and cleaned, then we need to be able to study it. We can study it using different techniques for different reasons. We can look at the DNA for genetic relatedness between bird species, or we can also study the parasites that might reside in the bird. But because there's such a small amount of DNA, it needs to be amplified so that we can detect it. So imagine a microscopic copy machine that takes the DNA and makes multiple copies of it exponentially. This is called polymerase chain reaction. This technique is used in most molecular biology protocols to amplify DNA. Once we decide what we want to study, we have specific probes that we use that will amplify the certain genes that we want to study. So after the PCR is completed, we put it in the machine, and this is where the replication continues. After this is done, then, we take the samples and run them on an agarose gel. And it's from here that we can determine which birds are infected. Once the gel has been completely run, we can then examine it using UV light, and this is basically what the outcome is. These dark spots here are called bands. They represent every individual bird, and you can see that if the parasite for avian malaria is present in the blood sample, it will show up on this gel. So these birds, of the 96 birds that were screened here, these birds that have dark bands are the ones that were positive.
Next, we're going to go to Dan Thomas. Dan is the director of Vireo, which is our collection of bird photos.
Hello, my name is Dan Thomas. I'm the manager of the Vireo collection at the Academy of Natural Sciences of Drexel University. Vireo stands for Visual Resources for Ornithology, but it's more than that. It's a collection of bird photos for anyone interested in learning more about birds. It was founded in 1979 by our then curator, Frank Gill, with a donation of photos from Crawford Greenwald. It included his stop-motion photos of birds in flight, including hummingbirds, which were taken in the 1950s, well ahead of their time. The Vireo collection is a unique complement to traditional museum specimen collections, and it's data-rich too. Just like the collections that Nate and Jason curate, it has data about where and when the birds were photographed. There are about 10,000 species of birds around the world, and Vireo has roughly 75% of those species represented in its photos. Over 100,000 of those photos are available on the Vireo website. What are some other ways Vireo photos are used? The photos are licensed for use in field guides, apps, textbooks, and other non-commercial uses like lectures and presentations made by ornithologists and bird enthusiasts around the world. As one of the first and longest running, the collection offers a historical perspective of habitat, bird distribution and behavior, and a historical perspective on scientific wildlife photography too. It also includes voucher records for new species and distributions in support of ornithological research. Here at the Academy, we like to connect people with nature in meaningful ways, and that's one thing I think Vireo does particularly well. Photos of living birds are incredibly educational and an incredibly powerful conservation tool that speak to people in ways specimens and research sometimes cannot. It can be particularly hard for someone to understand from looking at a dead bird why they should conserve habitat to save a particular species. But if you have a photo of a living bird living its life in the wild, it really connects with people, and they get it. If you'd like to see more Vireo photos, you can find them on great apps like the Audubon Guide apps or the Fieldstone Guide to Birds app and a field guide coming soon. The Stokes Field Guide to Bird also features Vireo photos almost exclusively. There's a Google Arts and Culture online exhibit called Flirtatious Feathers that some of you may remember from our past Art of Science exhibits. Of course, you can always find Vireo photos at vireo.ansp.org.
So we've talked about how we're using these specimens to study genealogies of birds and also to study the evolutionary history of birds and also their parasites and pathogens. One other really important aspect of our collections here is that we are not the only ones that use them. People come from all over the world and people borrow specimens from all over the world from our collections. So a specimen like this one might be used for work that we're doing in the lab, but it might also be used by researchers in Brazil or Peru or in Europe somewhere. And this is one really important aspect of museum collections. These are resources for researchers all around the world. The other thing that I think is really important to point out is that those of you that are bird watchers, you all are familiar with field guides. Well, those field guides were not just painted from photographs and from artists going out into nature. Artists come to our collections and use specimens like these to paint those plates. And those plates would be very difficult to paint without having access to specimens like these. Thanks for visiting the Ornithology Collection. We hope you'll come back in the near future, and we look forward to seeing you again.